This is the report on the project RHIZOTOX, an EPPN project between the Gregor-Mendel Institute in Vienna and the Forschungszentrum Jülich. RHIZOTOX is set in the background of allelopathy. Allelopathic plants are plants that produce and release secondary metabolites that are able to suppress the growth of neighboring plants. One risk that such donor plants run into is, though, that they intoxicate themselves by suffering from their own phytotoxins. This is a phenomenon called autotoxicity. We set out to investigate the genetic basis of autotoxicity, and we chose Barley as a model to do so because Barley is a diploid species with a good quality genome sequence. It has mutant resources available, and it is known to be an allelochemical donor, particularly because it produces and releases gramine and ordenine, among other compounds. Our strategy was to use quantitative trait loci mapping to identify genetic determinants of autotoxicity tolerance. So for this, we chose a population with trait variation and genetic diversity that has been partially genome sequenced already by our collaborators. And our goal was to collect phenotypic data on root growth under allelopathic and non-allelopathic conditions. In particular, we chose a collection known as Composite Cross 2. So this is material that has been provided by our collaborators Daniel Koenig at the UC Riverside in California. And it's a collection that is derived from 28 different parental land races that were intercrossed and propagated in the field for almost 60 years in a natural evolution experiment. At the Gregor-Mendel Institute, prior to the EPPN project, we gathered preliminary data by growing barley on a blue sheet paper suspended in a nutrient solution, such that we could 2D phenotype roots in high contrast. This is illustrated here. So the root grows along this paper, and we can measure different root architecture traits. The caveat of this approach is, of course, that there is no natural soil environment in which we can study allelopathic effects. But still, what we can do is we can apply different chemicals such as gramine, shown here, in varying concentrations to determine the toxicity effect on these barley plants. Prior to RHIZOTOX, we did this for the CC2 parental land races, which we grew on 5 millimolar gramine or in a controlled solution and measured root growth inhibition. And what we noticed was that among these land races, we got substantial variability in the response to 5 millimolar of gramine. Illustrated here, so this is a range from the most tolerant to the most sensitive land races along the x-axis. We can look at this in more detail here. For example, land race number 6 shows very little sensitivity to gramine, whereas landrace number 18 shows very high sensitivity to gramine. So we already knew that there would be phenotypic variability in these among these different genotypes. We then chose the rhizotron facility at the Forschungszentrum Jülich to conduct an experiment in which we could measure these effects in a more natural environment, namely soil, growing plants in thin layers of soil to be able to measure the 2D architecture of the roots. However, when the pilot experiment was run at the Forschungszentrum Jülich, we noticed that there was a substantial problem, which was that the solvent, DMSO, which we have to use to resuspend ordenine and gramine, caused damage to the plants when applied in soil, something that we had never observed before when we grew them in vitro cultures. So we were not able to overcome these problems in a reasonable amount of time, and we therefore had to change the strategy of the experiment. So in the end, we did not apply either gramine or ordenine, but had to conduct the experiment without treatment with allelochemicals. Instead, we decided to phenotype the root architecture of these parental land races to get an idea about the general variability of root phenotypes in this collection. So we ended up phenotyping 144 plants with 6 replicates per genotype over a period of 18 days. When we look at the data, then we see that there is a massive phenotypic variability among these different genotypes shown here along the course of the experiments. This is the time axis, this is the length of the main root, and this is divided by genotype. And one can see that there is almost a twofold difference between the slowest and the fastest growing genotype. We also notice that there was variability in biomass production. So what we are looking at here is shoot dry weight by landrace, and again we could see a 2 to 3-fold difference among genotypes in biomass production. So currently we are still performing more detailed analysis of these phenotypic data, and we would like to use these data ultimately for association studies between phenotypic and genetic variability. Fortunately, we had another EPPN project in this call, namely Project 180 PhenoHorde, in which we phenotyped the above ground tissue of these very same land races as well as some of their crosses, such that we can now combine the root phenotype data with the shoot phenotype data. I would like to give very special thanks to Anna Galinsky and Kerstin Nagel from the Forschungszentrum Jülich for being very flexible in project planning and also very efficient in its execution. Unfortunately, due to COVID-related restrictions, we could not carry out this project in person in Jülich. Instead, Anna and Kerstin took over and managed to make this a successful project despite the restrictions. I'd also like to thank Zane Duxbury and Patrick Kuta at the Gregor Mendel Institute for their input into the project. Thank you very much.